Morning, everyone. All right, so this is the last video that you will need to know for your exam, for your quiz, everything. And I'm sure most of you will be glad that it's over with. But it doesn't look that bad as far as all this writing on the board. But today we're going to talk about immunohistochemistry. And this type of technique is probably one of the most used techniques up in the lab, other than special stains. Special stains does have its uses, but this one has even more uses. I think maybe you saw that back when I was talking about connective tissue. One of the stains in the packets that they talk about is the PTAH stain. That's kind of being phased out, even though it's probably still used in some labs, but not as much because of this. Okay, IHC is much more sensitive. It's also much more specific in some cases. And so it gets the pathologists the answer that they need. With today's cancer cases and just all the different types of malignancies that we are dealing with in today's cases, they become much more complex. We're finding out that we're sending out more and more slides to do molecular testing on them because they can't seem to get the answer either through their H&E or through a special stain and or through this, okay, IHC. So, all right, so the definition of IHC is a technique for identifying cellular or tissue constituents, being the antigen, by means of antigen-antibody interactions. The site of antibody binding identified either by direct labeling or by use of a secondary antibody labeling method. So what we're going to be talking about today, the packet, and if you have the book mentions direct, indirect, IHC, and avid and biotin. Avid and biotin is used in conjunction with IHC. So, but in some cases, I guess you can use the antigen biotin by itself, which we don't normally do. Okay? All right, antigens. It's a molecule that induces the formation of an antibody and bears one or more antibody binding sites called epitopes. Okay? Composed of highly specific topographical regions made up of small numbers of amino acids or monosaccharides, units called, again, these are called epitopes. So this is, these are all over, obviously, the cell surface. So depending on what you're looking for is depending on, you know, what type of tissue. Everything affects this by, again, fixation, processing. So let's just break this down. So antibodies belongs to a class of serum proteins known as immunoglobulins. You guys should probably know this anyways, coming from your immunology backgrounds. Can be monomeric, dimeric, trimeric. Composed of two heavy and two light chains formed in the humoral immune system by plasma cells. The end cell mean the B lymphocyte, and that B lymphocyte transformation is after recognizing a foreign antigen. So depending upon, let's say, antibody specificity, antibody characteristics to bind selectivity to a single epitope or an antigen. Now I was telling you specificity, or excuse me, selectivity and specificity and sensitivity. Sensitivity refers to the relative amount of antigen that an immunohistochemical technique is able to detect. Example, a technique with a high sensitivity is able to detect smaller amounts of antigen than a technique with lower sensitivity. Okay. So also specificity, or how specific it is, falls under this as well. But if you're looking for something very, very specific, then obviously you're going to want your antibody to be more sensitive. Okay. Production of primary antibodies. All right, so we have two different types of antibodies. We got monoclonal and polyclonal. Polyclonal antibodies produced by immunization of an animal with a purified specific molecule, an immunogen, bearing the interest of the antigen. Animals will respond to the immunogen. Antibodies can then be obtained by bleeding the animal, okay, to collect the blood to get the serum. Animals will likely produce numerous clones of an activated plasma cell. And again, we're talking about polyclonals. Okay. Each clone will produce an antibody with a slightly specific or specificity to the variety of epitopes present in the immunogen. Okay. So basically, a polyclonal can attach itself to more than one epitope, is what they're saying. Monoclonal antibodies, a little bit different, made by combining plasma cells or B lymphocytes to produce a specific antibody with a neoplastic myeloma cell line. Okay? And once they get the cell line going, they can take the cell line and they can start cloning them. Okay, so by cloning the cell line can be grown and multiplied in cell culture or acetic carry my writing. Fluid. Okay, there's acetic fluid theoretically specifying to unlimited numbers. So you can go ahead and clone these cell lines and get multiple cell lines from them. Okay. The thing with this is that monoclonal antibodies obviously are more specific, okay, to one epitope, whereas primary or polyclonals are not. So these are going to give you very specific and less non-specific background. Okay. And we'll talk about background in a little bit, but okay, so these two types of antibodies, and now I think they've got one more, and I want to say it's a rabbit monoclonal antibody that you can actually do from a rabbit instead of a mouse in the cell line. But I don't exactly know the whole thing behind it and how they do it, but it must be similar to this. So they just do it in a bigger animal because then they can bleed it a few times, a lot actually. All right, so that's a little bit about the background of this and how there's two types of antibodies, the polyclonal and the monoclonal. Now, the types of methods that you can use with this is dependent upon the pathologist and or your lab. The one that we're going to be talking about the most is the avidin biotin complex. Okay, so there's 4 different types, and this is what I was talking about earlier. There's a direct method, and I'm not going to go into it. An indirect method, I'm not going to go into it. Soluble enzyme immune complex, or avidin biotin complex. Okay, now the avidin biotin complex is part of the whole process. It's not separate. It's not it's not used like a direct or indirect, or even an enzyme immune complex. But the avidin biotin complex is used along with it, and you'll see that in a bit. Avidin is used in this because it's an egg white protein that binds 4 molecules of the vitamin biotin. Okay, so it has a high affinity for it. That's basically what it says. So, has a high affinity for biotin. ABC method, the primary antibody is followed by a biotinylated. This is important. Biotinylated. So it's got a biotin tag hanging off of it. Secondary antibody. Then followed by an application of a preformed avidin biotin enzyme complex. And like I said, we'll get more into that. There's 2 types of visualization that you can use when you're doing immunohistochemistry. One of them is immunofluorescence, and the other is enzyme IHC. And enzyme IHC is the one that we're going to concentrate on. Immunofluorescence is okay, but the thing behind this is that you have to immediately read your slides. Okay, whereas enzyme, you don't have to worry about that, because when you develop it, and we'll talk about that too, I don't want to give too much away. When you develop it, it's insoluble. Okay, so it can never go away. You can never get it out of the tissue, is what I'm trying to say. All right, so enzyme IHC. The enzyme in the presence of a substrate and a chromogen, okay, produces the indicator system to visualize the location of the antibody on the cell, or in the cell. Enzyme that we use in the lab is called horseradish peroxidase. HRP, in the presence of hydrogen peroxide, the substrate, find a chromogen called DAB, 3,3-diamminobenzidine. A while back when we were using this, they said that it was a suspected carcinogen. Now they're saying it's not. So I guess it doesn't cause cancer in humans. But this chromogen will identify the sites of the antibody that you're binding to by forming a colored compound. And this colored compound is brown. Okay? In your reading, they may have talked about that if you are using something that's really sensitive, okay, that is very, very specific but sensitive to a certain epitope on a cell, then you may or may not be able to see the staining that well. The staining will come out, but it'll come out light. So there is a way of intensifying that color. And the way of intensifying that color is by using something called nickel chloride. Okay? So, again, if you're using a antibody, and I'm just going to AB prime, an antibody that is very specific you might want to use an enhancer is what they call it. Oops. Telling you. Enhancer to the DAB component. Alright. So what it does is it turns it black. Alright. So when you see it, you're going to see it. Okay. It's going to be much darker. You'll be able to see it because there might just be a few cells that are very specific for this antibody that is going to pick it up, and you're not going to see it with the brown. But if you put this enhancer and mix it up when you're making up the DAB, then you will be able to see it when it develops. Okay? All right. Some technical considerations. Okay, one, again, coming back to haunt you again, fixation and processing. All right? Formalin. Okay? I briefly talked about this, and I said it when we talk about IHC, then we're going to be coming back to it. Well, here it is. Formalin. Non-coagulating additive fixative that causes cross-linking. Okay. If you take anything away from anything in this from histology or the histotechnology portion of it, you will take away this, and you'll probably remember it for the rest of your life. All right. So formalin causes cross-linking of proteins. Okay. Methylene bridges are formed. They mask the epitopes of the cell surface. So here, cross-linking creates methylene bridges that mask the cell's epitope, which prevents the antibody from binding. Okay? So check to see if the tissue has been overfixed by staining it with Vimentin. Vimentin is in all tissues. So if you want to see whether or not you have cross-linking, then you take some tissue, it doesn't matter what it is, and you fix it, and then you try to stain it after it's been processed and embedded and cut and all this jazz. Progressively, you want to see if it's progressively lost as the length of time in the fixation increases. All right, so if something's been overfixed, okay, then try a piece of tissue that's been overfixed and try to stain it with Vimentin and see whether or not you get it. Supposedly, you'll progressively lose more staining as you leave it in longer. Okay? Because the longer you leave it in formalin, other than, formalin breaking down and you're getting pigment, okay, you will also lose your staining the longer you leave it in. All right, so formalin is one form of fixation that you can use, and it does obviously cause a little bit of problems, but there's ways around it, which we'll talk about. Zinc formation. Okay, it's really not zinc formation, it's zinc formalin, excuse me. Zinc formalin preserves immunoreactivity remarkably well. Okay, It's a great antigen retention and preservation of the tissue. Also, nuclear detail is extremely crisp because zinc salts, that's what they do. But the one thing that I didn't put on here, so we can add this, I'll add this to the list, is for, it also prevents cross-linking. Alright? So it prevents cross-linking. Another fixative that's out there called glyoxal, and that is kind of, I think that's more mentioned in the book than in your packets, but it's not going to be on the test. But I'm just letting you know that there is another one out there called glyoxal that can also, that's also safer than formalin, and it is a dialdehyde. Okay, but not in the terms, you can still, I'm pretty sure you can still use glyoxal for PAS, but the main gist here is if you want to use another one other than zinc in your formalin, then you can also use glyoxal. But zinc formalin also prevents that because of the zinc. All right, epitope enhancements epitope enhancements or retrievals. There's two of them. There's the HER and the ER, or however you want to pronounce it. This stands for, obviously, heat-induced epitope retrieval. Another name for it that they still use today is called antigen retrieval. A lot easier to say. Enzyme-induced epitope retrieval is not widely used, and the reason why it's not widely used is because enzymes tend to chew things up. That's what you want them to do. You want them to chew up the bonds, because these bonds are very strong. They're covalently bonded. So you need something, you need a lot of energy to break the bonds, right? So heat here is used to break the bonds, and enzymes are used to break the bonds or chew them up. The problem with this is that if you use too high of a concentration of enzyme and you leave it on there too long with your cells, guess what's going to happen? They're going to break and chomp through the cross-link, but then they're also going to get to your cells and they're going to chew them up. So not a good thing to use. And you couldn't use it in a histopathology lab anyways. You would probably use this more in a research lab. Okay? More this way is used more for research purposes. Well, all right. All right. So here we go. So heat-induced epitope retrieval. A couple ways of doing it. Pressure cooker is one of them, or you could also use the microwave, or you could have a machine like we do upstairs, and it does it for you. It heats it up to a point. If you have to use antigen retrieval, now some epitopes doesn't require it. So depending on, we have like a lot of different protocols in our computer upstairs that is attached to these machines, and if it has an antibody that requires antigen retrieval, then of course, okay, fine, then they're going to use it. And so the machine will heat up, because the machine does everything. It deparaffinizes, and then it does this. So it deparaffinizes the tissue, then if it needs antigen retrieval, that's the next step. Then there's different, if you're gonna be using a pressure cooker or if you're gonna be using a microwave, then there's different solutions, citrate buffer being the most widely used, for your solutions. And you put the slides in it, you heat it up to a certain temperature, and then just continue with the protocol. Sodium citrate buffer, like I said, is the most widely used. The pH is of 6. Like I said, on today's IHC machines, we have the Ventana Ultras upstairs. And this does everything. Like I said, deparaffинizes, does an antigen retrieval. Most conventional IHC automated machines automatically do these steps, as well as deparaffinize the tissue. All right. So, once you get to these steps, now, when you're preparing your slides to be doing this, okay, you need controls. This is a must. This is CAP regulated. So you have to run your controls. Now you've got two types of controls. You've got positive controls and you've got negative controls. Okay? Positive controls must be run along with each antibody. So for example, if you've got a case that needs P63, or excuse me, P16, which is a cervical, an antibody that detects cervical cancer. Okay, let's just say you have P16. You have to, and then let's say you have 3 different cases. Okay, the pathologists, let's just say one pathologist orders it on one case, and then you have another pathologist that orders it on a different case. So you have to, for each one of these cases, you've got to put a positive control on the slide along with the patient tissue. Okay? So you want to make, this is the way it usually looks like. So if you have a slide, okay, these are done on positively charged slides because of the type of environment the tissue is going through when it's going through the machine. So it's a lot of heat and a lot of chemicals and what have you. So up here, let's say we'll use a different color. Up here is going to be the control slide. So this is the control tissue.